I am Manomani from Department of Microbiology. Today we are going to discuss about dengue viruses. Detail about introductions about dengue viruses and what are the symptoms and causes of dengue viruses, and what are the techniques are used to diagnose the particular viruses, and how to prevent these diseases from the viruses. So, this is about the small introductions about dengue viruses. So, first the dengue viruses is a mosquito borne arboviruses which was belonging to the family of Flaviviridae and the genus Flaviviruses, and these dengue viruses is one of the vector borne viral diseases, and these virus can affect globally approximately 400 million people per annum. These dengue viruses can cause dengue fever in human populations. There is no specific antiviral treatment for this diseases, dengue fever for these diseases. The management and prevention measures to be taken by us is the only treatment sources for this dengue viruses. So, these dengue viruses can cause a mild fever, that is mild febrile illness to severe to dengue hemorrhagic fevers and dengue shock syndromes. These dengue hemorrhagic fevers and dengue shock syndromes can occur during the secondary infection, and this is the most deadly dangerous nature of these diseases. So, what are the causes of dengue? So, dengue, which is belonging to, we already discussed, dengue viruses is caused by the dengue virus belonging to the Flaviviridae family, and in this family dengue virus have 4 serotypes. So, dengue virus 1, dengue virus 2, dengue virus 3 and dengue virus 4. So, infection with one serotypes which give a lifelong immunity only to that serotype. For example, dengue virus 1 causes dengue fever someone person, and these viruses can give immune power, that is lifelong immune power to that particular dengue virus 1 only. So, what are the symptoms of dengue viruses? These are symptoms only for mild to severe fever, headache, back pain, bone pain, vomiting, eye pain, muscles pain, joint pain, high fever, and also tiredness, weight loss, also some other symptoms for these viruses. So, how does dengue can spread? So, dengue viruses can spread through a female mosquito named as Aedes. So, Aedes, which is primarily infected a person with dengue during the viremic stages. Viremic stages mean the person suspected or infected with this viruses which was circulated in their blood. At that stage only the viruses became easily transmitted from one person to another persons. So, after that the virus can multiple or replicate inside the mosquito for about 8 to 12 days. The infected mosquito can then transmit the viruses to another healthy person through its bites for the rest of its lives. So, the Aedes mosquitoes mostly active during the daytime only, particularly in the early morning and late afternoon. So, what are the lab techniques which was available for the detection of dengue viruses is, first one is for the direct methods. So, direct detection methods which include NS1 antigen test and RT-PCR method. So, for this NS1 antigen test which is used to detect the viral proteins in the early stage of infection, that is the infections can, when persons are infected with these viruses, within 5 days we can collect their blood samples and then detect it. Then next one is RT-PCR. RT-PCR we can easily also viral RNA. NS1 is a which was used to detect the viral protein. RT-PCR methods are used to detect their RNA particles. So, second one method is indirect detection method. So, indirect detection methods are mostly used for serological examinations for this only. For this serological test, we use 3 different types of tests. First one is IgM ELISA test, IgE ELISA test and heme agglutination incubation test. So, after exposure of 5 days or after examination of these viruses, we can easily find the infection through the IgM ELISA. For IgG ELISA technique, secondary inversion of dengue viruses, we can easily predict that particular viruses with the help of IgG ELISA technique only. Heme agglutination incubation techniques, these techniques are used to detect the antibodies for the correspondent antigens, and these heme agglutination incubation tests is now commonly used to find the viruses. So, what are the preventive measures behind dengue viruses? So, first one is to eliminate the mosquitoes by using larvicides or indoor spraying. So, water is the main sources for this viruses transmission. So, through this water the larvae or mosquito can lay the eggs inside the water. At the stage the larvae we can easily spread from one place to another place. So, first we should eradicate the stagnant water like flower pot, tanks, tires. So, we should eliminate the stagnant water. We can spray larvicides on it. So, personal protections, use mosquito repellents, nets, long sleeves clothlings can be used. Stay indoors during the bee biting times, for example morning time or afternoon session. So, for communal measures, we will create any awareness programs among peoples. For vaccinations, so only dengue vaccine is the one and only choice of vaccine for this particular viruses, and these vaccines can only approved in for some countries and some infected individuals or persons only, not used for all persons and all countries. So, for environmental measures, so for environmental measures also we can eradicate the waste, we can dispose of waste in proper way and eliminate the stagnant water, close all moisture containers, and then the next one cover all the open water savings like containers, barrels, tanks are tightly covered with a lid, and also disposal of solid waste, and we can easily prevent the spreading of this kind of viruses. Thank you.